So next up, we're going to move into our next session, which is highlighting progress in drug development for CMT. Our first speaker will be Bradlee Heckmann, co-founder and president of Asha Therapeutics, and he will present on targeting SARM1 and axon degeneration as a disease-modifying strategy for CMT2A.
Well, thank you very much for the warm introduction, and of course for the invitation to be here today. I barely made it in actually with the rain out there. It got a little delayed, but nonetheless, I'm thrilled to be here. First of all, I want to preface my talk with I am not a CMT expert. I'm actually originally a molecular pharmacologist by training, and I am learning slowly but surely the ins and outs of CMT pathology and how hopefully we can make a difference with some of the novel therapeutics that we're working on at Asha to be able to get there. And of course, I have a bone to pick with the organizers. You put me after Steve, the godfather of the field. So I have big shoes to fill, and hopefully I can do it at least a little bit of justice. So when we think about axons, right, that's one of our major foci, if you will, at Asha Therapeutics is on neuronal integrity. And from a metabolic perspective, so mitochondrial function. And so I think you can probably understand where I'm going with this when it comes to where do mitochondria influence cell function in the context of CMT, and that's most likely going to be in our axonal degeneration forms driven by mitofusin 2 mutations, so CMT2A. And so I'm not going to give any background really on the two forms. You all know this better than I do, but we will be focusing on CMT2A in particular with mitofusin 2 mutations driving this pathology. And hopefully by the part of the talk where I get to the therapeutic intervention, you'll see why we are doing that. And so when we think about these different forms of CMT, and in particular mitofusin mutations that are altering mitochondrial dynamics and altering mitochondrial function, we know that this is really a yin and yang balance. And so this is an area that affects, of course, every cell that has mitochondria in our body when it comes to energetic maintenance, homeostasis, prevention of cell death signaling. And so this is a tightly regulated process. And so going either direction, whether that's towards hyperfusion, right, making these big mitochondria, or towards hyperfission or fragmentation, where you have a bunch of smaller mitochondrial fragments, both of those can, of course, be bad to overall health and well-being. And we know that in the context of a lot of neurodegenerative diseases, right, everything from our more typical AD, PD through our neuromuscular diseases like ALS, there is a significant role that mitochondrial dynamics and the regulation of these pathways play in disease pathology and pathogenesis. And in the context of CMT, there's not, especially CMT2A in particular, there's not really any difference there. We know that in the same context, these dysfunctional mitochondria and the inability for these mitochondria to distribute properly within neurons ultimately leads to a host of signaling pathways, one in particular that we'll be discussing today, that lead to axon degeneration and ultimately cell death at the level of the whole neuron. And so this has been shown multiple different ways, but the rat models, obviously mouse models of disease where mitofusin 2 mutations cause axonal degeneration and mitochondrial dynamics dysfunctions, including mitochondrial clustering, decreased fusion of those mitochondria, and ultimately fragmented states depending on the mitofusin 2 mutations. And that, there are a variety of methodologies to try and restore that mitochondrial homeostasis, including things like overexpression of the mitofusin proteins, as well as a host of other avenues. And so we were looking at this in a very different way. And realistically, it all comes down to one person who's in the room today, Dr. Aaron D'Antonio, who suggested when we were looking at our drug in the context of ALS pathology, he brought it to our attention. He said, one thing that we studied was the role of this protein SARM1, which of course he and his colleague Jeffrey Milbrandt really characterized and discovered this function in axon degeneration. And he said, we've seen if we knock out SARM1 in models of CMT2A, we see rescuing of the phenotype. And so before we get to a little bit of the data that they generated, you first need to know a little bit about SARM1 and why did I go through mitochondria into SARM1 biology. And the reason is that SARM1 is heavily regulated by the ratio of key mitochondrial metabolites and energetic substrates, in this context, NAD and NMN. And so these two substrates ultimately have a balanced ratio, and when that gets skewed, you ultimately lead to the activation of SARM1. You lead to obviously changes in hydrolysis of NAD. This activation where SARM1 goes from an inactive closed conformation into this dimerized activated state where you have this NADase activity of the protein, and ultimately this results in signaling leading to degeneration first of the axon, and then ultimately propagates this whole mitochondrial dysfunction towards the path of cell death as a whole. And so you end up with more broad neurodegeneration as a consequence of this. And so there's this direct interplay between mitochondrial function and SARM1 activity, and then a feedback loop between SARM1 activity and mitochondrial function as a whole. And so when these two, they essentially reinforce one another, driving further pathology downstream. And so what Aaron and Jeff really showed in the rat model, the CMT2A rat model, the H361Y MFN2 mutation model, was that if you knock out SARM1 globally, now mind you, this is a germline deletion of SARM1, you were able to normalize or trend to normalize mitochondrial integrity and mitochondrial parameters, right? So you're starting to restore that upstream mitochondrial phenotype. But more importantly than just restoring the mitochondrial phenotype that was caused by the MFN2 mutations, they saw very nice reinnervation occurring of the neuromuscular junction, as well as improvement in key readouts of actual behavior and pathology, in this case, a grip strength analysis showing here that you see a significant improvement in the animal's ability to have coordinated motor function at the level of at least grip strength. And so what does this really suggest? It suggested that SARM1 could potentially be a very viable therapeutic target for intervention that could do essentially two things, right? It's going to obviously stop this SARM1-driven axonal degeneration phenotype while supporting or at least trying to support healthy upstream mitochondrial restoration, which we know that in anything in neurodegeneration, blocking cell death alone, what does it matter if we have a dysfunctional neuron sitting there, right? It's really a two-part question in the fact that we want to not only keep those around, but we want to keep them as functional as possible towards a homeostatic state. And so hence the importance of restoring mitochondria in the process. And so when we were developing out, we saw SARM1 as an excellent target in other diseases, including ALS, as well as drug-induced neuropathies that have a very, very similar mechanistic pathway involved through to SARM1 activation, just not driven by MFN2 mutations, but essentially the same level of mitochondrial dysfunction that's occurring. And so we used a technology that we developed at Asha. It's a drug design platform that we call PRISM, and we developed out a series of SARM1 inhibitors that function through a unique mechanism of action. And so if you look, you can see that SARM1's really on this side of the equation with axon degeneration, and this inactive conformation of SARM1 is what we really want to keep the protein in. And so a lot of other attempts have been directed at SARM1 inhibition, including Aaron himself, which uses a different inhibitory approach targeting the actual NADase domain that will form here between these TIR domains upon dimerization when SARM1 becomes activated. And so what we ended up doing is taking a little bit of a different approach, and we use what we call an intramolecular glue. So this is a small molecule that binds to a pocket within SARM1 and essentially glues the protein into this inactive conformation. So it doesn't open up and dimerize to form those NADase domains that will, or the NADase active site to hydrolyze NAD, and so thereby blocking its function. And so we developed out a whole series of molecules. I'm showing 4 of them here out of the series that we designed. And you can see to varying degrees, we saw inhibition of SARM1's activity in a test tube. This is an in vitro NADase activity assay. And one of them in particular, this G6036 compound, stood out as a pretty robust inhibitor with an IC50 value of right around 132 nanomolar. And so in addition to other assays, especially in the pharmacology space on the ADME and PK of the molecule, we ended up nominating this 6036 compound as Asha 624. And we saw, sorry, get back there. We saw a very robust inhibition, like I mentioned, in the NADase activity, but we can also treat iPSC-derived neurons that have been stimulated to activate SARM1 with either a more general mitochondrial activation through rotenone exposure or a more specific activation of SARM1 using VACOR. And so in both instances, there was a dose-dependent response and protection of these axons when treated with Asha 624 as a whole. And so we ultimately took this further from cell studies. We put it into animal models. In this case, it's an ALS model that has SARM1 activation, very clear locomotor pathology and loss of innervation at the level of the neuromuscular junction. We treated these animals after they had established locomotor pathology and motor dysfunction. And we saw that after about 12 weeks of treatment on compound, treated animals had a significant improvement in their overall stride parameters. So this is, in this case, stride length, which is one key parameter that's normally dysfunctional in this ALS model, whereas vehicle-treated animals continually digressed as their disease progressed. We also saw very robust improvements in grip strength, as well as general motion in a general locomotor activity in an open field analysis. And we also saw at the level of the neuromuscular junction that when we treated with compound, we saw a very nice maintenance of the neuromuscular junction architecture, right, which is normally becoming fragmented in these animals. And we saw a nice improvement in the innervation of the NMJ. So very consistent with what Aaron and Jeff and team showed in the CMT2A models with the SARM1 knockout with this reinnervating phenotype, we can see in the ALS models, and we also saw some improvement in other parameters as well. We took this a step further and said, okay, that's great. It's a model of ALS pathology, but what about a more direct activation of SARM1 in the context of a drug exposure? And so chemotherapy-induced peripheral neuropathy and these taxane and platinum-based chemotherapeutics in particular are well known to activate Wallerian degeneration and cause, obviously, neuropathic pain. And we did a very similar study where we treated animals either prophylactically or after establishment of the neuropathic pain. And as you can see pretty clearly in this graphic, we saw significant improvement after establishment of symptoms, and those that were prophylactically exposed to the compound really never developed the neuropathy at all. And so again, another example of the efficacious inhibition of SARM1 being a potential therapeutic avenue where you have this Wallerian degenerative phenotype being present. We obviously have looked at a variety of other contexts of the drug, including safety, both acute and chronic safety, all preclinically to date. We're hopefully going into clinic in the first of the year. And we saw no unremarkable level of safety issues that came up with the compound. We can see that there's no major issues on any key blood parameters. We saw no major organotoxicity, no real genotox, and we had a pretty clean CYP profile for your key pharmacology parameters. So the benefit of this is that we are now approaching what we believe to be almost a clinical stage asset in terms of moving this forward into clinic. And so how does this all apply to CMT? Well, we've been very, very thankful that earlier this year, a few months ago, grant was sponsored by the CMT Research Foundation to take this and actually look at this both in vivo and in vitro and iPSC lines from human patients with mitofusin 2 mutations, as well as obviously the animal model of CMT2A. And so I'm happy to report we're now started dosing in our iPSC models. The animal model will be coming up in the next few months here. And so hopefully this time next year, we will be able to give a full report. And we're really looking forward to, as we move through our first phase 1 trial on safety with the compound, looking at how we can engage and assuming we see positive results like we've seen in other models preclinically with CMT2A, how we can move forward rapidly into a patient population with the disease and try to make an impact there as well. And so on that note, I think I am right on time. I'd like to thank the entire Asha team, as well as my academic laboratory who does some of the work for us as well. Obviously, Jeff and Aaron for bringing this really to our attention, and Aaron is the key there into the context of CMT2A, to the CMT Research Foundation, and of course to the patients and families. We are really hopeful that we may be able to make a small dent and hopefully make some impact here in the very near future. And so on that note, I thank you. I'm happy to take any questions. And yes, thank you. Except from Aaron. Aaron can't. No, I'm kidding.
Very nice presentation, really very nice. You only showed us total knockouts. And with SARM, it's always interesting. How much do you need to knock it out? And the reason why I'm asking, I wasn't able to follow the pharmacology, but you had 2 mg per kg as a very efficacious dose, 100 nanomolar as the IC50. So I'm just trying to, from a drug development perspective, where do you think you have to be, as much as you can say?
Yeah, so that's a really good question. And so that's been one of the discrepancies between genetic models, right? A germline knockout, and a lot of the previous studies that have been done using some of the base exchange inhibitor, SARM1 inhibitors, is that, you can knock out SARM1 in a model, you see a very robust phenotype, you can treat, you still see a positive impact, but to a differing level. And so one of the things that we see with our molecule is that because of the mechanism of action, where we're actually gluing this into a complete inactive state, what is known, at least to date, and Aaron, I'm sure, would love to comment more. He knows way more in depth on SARM1 in this context. But when you have this conformational change of SARM1, where it undergoes its structural transformation, at least to date, that transformation is required for all functions of SARM1, whether they're the NADH dependent or independent functions. So we're hopeful that the molecular glue that's blocking that transformation and not just blocking the NADH activity is more hopeful to be more of a mimetic of a genetic knockout than just an inhibition of the NADH activity.
Question from the online. Has been identified which domain of SARM1 Asha 62 binds to, the TER domain or the ARM domain?
So that's a great question. So we hit a unique pocket that is in the ARM domain of the protein.
And then there's another question. Can the drug penetrate the blood-brain barrier? If possible, how much brain plasma ratio?
Yeah, so we do see pretty robust penetration into the CNS. I do not remember off the top of my head the ratio between plasma and brain, so I apologize there. But we do see very good penetration, and we have good tissue kinetics within the brain, keeping levels for approximately 9-hour half-life in the CNS. Yes.
Hi, Brett McCrae, University of Michigan. Great talk. It's really exciting work. I'm curious, it's a hard question, I think. So SARM knockout has been protective in some models of axonal degeneration and not in others. Any insights into why that might be? Is there possibly a hint that maybe mitochondrial dysfunction, it needs to be part of that axonal degeneration phenotype for it to be effective?
Yeah, that's a really good question. So you're absolutely right. There are models, and even in the ALS space, models of where you can knock out SARM1 and have varying degrees of efficacy of that, right? And even coming down to certain types of parameters, right? Part of that, we go back to the aspect of the NADH dependent and independent functions when it comes to the pharmacology interventions. When it comes to a genetic modulation, there is a potential that mitochondria play a larger role outside of just obviously activating SARM1 through NMN-NAD ratio changes. One of the things that's interesting is if you take the model that we use for ALS, for example, this TDP-43 model, that TDP-43 pathology activates obviously a host of mitochondrial impairment as well, right? So it's mimicking essentially what you'd see with MFN2 mutations, obviously a very different mechanism to that point, but nonetheless, the mitochondria are playing a significant role. There's also an implication of potential immune changes as well. We know that it's a TLR-like protein, and so there's also potential that you're altering immune responses, and Jeff and Aaron are doing work now on a different set of investigation in the context of macrophage and microglial biology as a whole. So there's quite a potential that you have more than just that axonal degeneration phenotype that plays into that. Yes.
Yes, great talk. Srikanth Ranganathan, Program Director from NINDS. Couple of questions. Maybe you did answer this. Is that binding site conserved across species?
Yeah.
And secondly, if this compound or the small molecule, is it only affecting the degeneration, or can it be also for recovery or regeneration?
Yeah, that's a really good question. So first of all, the site that we're hitting is conserved across rodent, canine, and human. So we don't see any issues with the compound. We see activity across all, at least of rat, mouse, dog, and human. The context of the second part of your question. Sorry, what was the second part? I was thinking about his question actually earlier.
Question, but whether it's only stopping degeneration or can it have
Yeah. Okay. So what we believe is happening, right, is that you have probably a dual effect that's occurring, right? We're able to take inactive SARM1 and keep it inactivated, right? But we also hit activated SARM1. So when it comes to a degenerative phenotype, we think that we're obviously stopping that degeneration. When it comes to regeneration, there's a whole nother aspect of that. SARM1 activation does have a tendency to downregulate pathways that are involved in neuronal or axonal regeneration. But we've not tested directly if we have an actual regenerative phenotype occurring. We could argue that based on some of our data, we may. But again, that's a very fine line to say, we were actually able to regenerate an axon with it. But the plausibility for it to happen is definitely there. Perfect. Thank you all.